International Union of Physiological Sciences to really deal with issues of complexity and modularity and reproducibility and so on. And then I'll get down to some of the details around the IT infrastructure that we've been developing for handling reproducible modelling. So to start with, computational physiology, and this involves being able to deal obviously with events at the molecular scale, and the case I'm going to talk about is the heart as an illustration, but it's equally applicable across any organ system. But just as important as the molecular behaviour is the behaviour at the cell level and tissue level, and very often 3D tissue structure becomes critical for achieving the phenotype of the organ, the function of the organ. So what I want to talk about really is the process of being able to use engineering and physics to try and develop models that deal with these scales and apply the laws of continuum physics at the level of tissue properties, and deal with the fact that you often have multiple types of physics going on, activation processes, mechanics, coronary blood flow, fluid flow in the ventricles, and also look at how that goes wrong under certain circumstances with arrhythmias, but also how these models are applied in a clinical setting where very often the source of the data becomes imaging, MRI or CT or ultrasound data, and the models can be used to help gain insight into clinical situations. So from an engineering perspective, it's absolutely critical that we have the ability to really understand structure. So we've developed tissue structure. So we've developed instruments where we can move a block of tissue between a milling head, precision milling head, and a confocal microscope, so we can zoom in through 4 orders of magnitude down to the level of individual cells, and then look at the collagen orientations in these 3D tissue blocks, and then out of that extract the density and the orientation of collagen that we can then characterize with the constitutive relations that we define for the tissue when we're solving the physics for say, mechanics. So it's very important to gather data and to be able to characterize but also understand the variability that exists in normal physiology and also in pathological conditions. Likewise, we need to be able to measure material properties, and often we want to understand those material properties in terms of the underlying mechanisms across from molecular through cellular scales, but often we need to develop instrumentation to be able to directly measure the material mechanical properties in this particular case of the tissues, which are always anisotropic, that is, they differ in different directions. It's always inhomogeneous and it's always nonlinear. So we've got to be able to characterize the orthotropic mechanical properties of these tissues. And I think this also illustrates the fact that the development of new instrumentation to make the measurements is just as important as the modelling, because often your ability to understand the physiology starts with your ability to make measurements of properties, and then you interpret that through the models. So here, for example, is an instrument that we've developed over many years to be able to measure the electrophysiology, myofilament mechanics, metabolism, signaling, etc. in heart tissue preparations. In this case, a trabeculae, a little muscle taken from the inside of the heart wall, and then mounted in a device where we can subject it to all sorts of measurements and then interpret those measurements with mathematical models. Then the net result of that, of dealing with the geometry, material properties, and linking down to molecular behaviour, is the ability then, using finite element techniques, to be able to pull that all together to look at the overall function of the organ, in this case the heart, and interpret the behaviour in relation both to cell and molecular level function, but also the underlying tissue structure, which is shown morphed onto the back of a finite element here in the inside part of the wall of a deforming heart. These arrows are just showing the principal strain vectors of the deformation. So once you have these models and once you understand the variation under normal circumstances, that it gives us the ability to then look at disease processes which often for cardiac diseases, or particularly for heart failure, involve very significant changes to the tissue organisation. So the tissues become much less able to shear and they lose their mechanical compliance in certain modes, and with the consequent impact on pump function. So obviously we need to understand it at the molecular level in terms of what's causing these collagen changes, but we also need to be able to characterise what the tissue, how the tissue has changed under these disease conditions. So the Physiome project now is really addressing all 12 organ systems of the body, and trying to use, many people around the world contributing, but using the same approaches of developing the instrumentation and being able to do the multi-scale modelling, usually with the aim of trying to apply these models in a clinical setting. And I'll show you a couple more projects to do with the lungs and the musculoskeletal system, but they are all using a similar framework of being able to handle the description of the mathematics at the cell and molecular level, coupled through to the tissue level and whole organ level. So this is the example I gave of the heart work, where we're modelling both the physics at this level and the tissue level, and also linking to the torso to be able to look at, for example, current flow out of the heart into the skin surface for ECG interpretation. But all of this driven by cellular processes that in some cases link down to genetics, and in particular with the electrophysiology linking down to the ability to look at differences and mutations of particular ion channels. So another example of this is work on the lungs being led by Meryn Tafai in Auckland. So she's using the same frameworks but applied to both the airways of the lungs and also the vascular system, and models of gas exchange at the alveolar level, but then being able to look at particularly perfusion ventilation ratios and understand the impact of particular diseases that lead to embolism, COPD for example, and then help and use these models, use the spatial distribution that can be measured in the clinic together with the models to help guide therapy for disease patients. Similar approaches in the musculoskeletal system. This is work being led by Tor Bezier at Auckland, where the models have the muscles and the bones capture, the 3D geometry captured, but the ability to then deal with phenotype or function at this level, depending on the particular question being asked, the appropriate muscle groups are involved. So one way of viewing all of this is to say we've had this wonderful compendium of parts that comes from molecular biology, 50 years of understanding the genome and the proteome associated with that, and metabolome and so on. We have the beginnings of being able to understand the different functions of the 300-odd cell types in the body, the mammalian body. But physiology really emerges at this scale here, typically a millimetre in one direction and typically 200 or 300 microns diffusion limited in the other dimensions. So whether it's a nephron in the kidney or liver lobule, etc., or osteon in bone, acinus in the lung, there's always a unit that's typically of that dimension where the structure, the 3D structure really matters for the physiological function, and typically there's only 3 to 5 cell types involved in determining the phenotype at that level. So one of the challenges is being able to develop instrumentation, as we've done for cardiac muscle, but for all these different types of functional unit that exists at that scale, in order to really be able to characterize the multi-scale function at that level. And then it's a relatively straightforward process of assembling maybe 500,000 nephrons into a whole kidney, etc., through branching 20 generations of branching blood vessels. I mean, the difference between a mouse, a man and an elephant in terms of renal function is the difference between them having 100,000 or closer to 1 million or 10 million nephrons. The nephron itself is common, so there's very little change in these functional units across mammalian species. It's just how they're connected together to deal with the function at the whole body level, depending on the size and environment that particular species lives in. So our challenge is to be able to use the enormous depth of knowledge that's coming out of the molecular biology community here to be able to link through to developing physiological function at this level and then up to whole organ models using pretty standard engineering technologies. And assisted, of course, by the fact that we've got these wonderful imaging technologies that allow us to look in on all these different scales, from MRI to micro-CT to confocal microscopy and so on, down to electron tomography and X-ray diffraction. But we also have 150 years of the wonderful development of the equations that describe the physics of the environment that these molecular processes operate in. So we owe a huge debt of gratitude to basically to the forefathers of continuum physics, who've developed these systems of equations where we can nowadays solve fairly readily, whether it's fluid flow through Navier-Stokes, finite elasticity, or Maxwell's equations, or reaction diffusion, that numerical techniques exist now to be able to solve these on these realistic 3D anatomies. The challenge is, how do we dive into the parameters that exist within these tissue-level equations to understand how the molecular cellular structure feeds into the properties of these constitutive coefficients at these levels. So, for example, in a reaction diffusion process in the heart, we want to know, how does the diffusion tensor depend on the underlying tissue structure and underlying material properties of tissues? How does the current source depend on the molecular properties of ion channels that feed that current into the tissue level to give you a propagating wave of activation in the heart, and so on? In this case, the structure of the constitutive relations that define the stress-strain relations for tissue. So, in a sense, the bioengineering challenge is to dive in to these multi-scale wormholes that link molecular behaviour through to the continuum level. So all of this involves enormous complexity of mathematics, and the danger here is that we just get lots and lots of groups working on different models using different ways of defining their terms with very little connection to each other. So we have to, as a community, I think, we have to develop a framework for being able to handle this level of complexity, which means that we need standards and modularity. We have to have open source standards, associated open source, freely accessible software and databases, and we need to encourage the development of a modular approach that allows people to reuse work that's done by other people. So I just want to talk a little bit about that framework and where we're heading with this. So we developed this markup language called SalML. It's an XML markup language, and it's fairly mature now, and we've got a worldwide community that contributes to this, and there's a fairly substantial database now of 600 or 700 models of a whole range of physiological processes that have been put into these standard XML forms, and that means you can derive the mathematics or the code or the interpretation, the annotation and so on directly from these databases. And it allows you then to be able to more easily plug someone else's model into your model to be able to take advantage of work done by others. So a lot of the framework now is in place for this. There's a similar framework for handling spatial variation using another markup language called FieldML, but it's not as mature as the SalML environment. And then we work with the SBML community, who are dealing more with reaction systems for molecular systems biology. So the annotation, the syntax is handled through MathML, standard XML description of the mathematics, together with certain conventions around how we represent model structure. But the semantics is handled through the standard set of ontologies that the various communities have developed in order that we can label the mathematical terms appropriately with Chebby IDs or Go terms or whatever it is. So these ontologies become accessible through the interfaces in order to then annotate the parameters and variables in the models in order that you can then reuse those models in another context. So here, for example, is a particular model of the respiratory chain. This is what the XML, a bit of the XML might look like, and the mathematics that's then able to be generated directly from the XML, the code that's available if you want to just copy and paste code directly into your own simulation codes. Or we've also developed an open source CellML simulation environment which allows us to read directly from the model repositories directly into the desktop software, or you can develop a model, your own model on the desktop, and then upload it into a workspace in the repository. So these tools now are becoming reasonably sophisticated in terms of being able to deal with modularity, reproducibility, annotation, and increasingly also analysis of the results of the modelling efforts. So one of the projects underway at the moment is now because all the models in the database have come from peer-reviewed publications, and incidentally, just to reinforce the point made earlier about open science and the lack of reproducibility, of the 600 or 700 models in the Physiome database based on CellML, I think about 10 were able to be coded directly from the paper without errors. So all the others, we had to go back to the author to find missing information or inconsistency between the equations, the figures, or the code. So it's no use just depositing code on websites. You have to have the models properly curated, annotated, and available in a way that guarantees that you can reproduce the figures in the papers. So part of the idea now is to be able to create databases that have all the transporters, all the ion channels, and then have models of those that make use of templates that deal with particular types of mechanism. For example, many transporters will have the same enzyme undergoing a 6-state cycle through the between ligand binding inside and outside. So we can reuse a lot of common mathematical structure for capturing phenotype, and then just have different parameter sets for different types of transporter. So here's one example of a sodium chloride cotransporter which is on the website, and you can access the UniProt IDs for that particular protein, access through looking up terms in Go, etc., in the Foundation Model of Anatomy. You can find and other databases, you can find where those particular proteins are expressed in different tissues, the actual structure of the model, the mathematics. Then I think the modularity issue is really vital because we are dealing with extremely complex systems. So I just wanted to illustrate one aspect of that for the cardiac case. If you have a cardiac myocyte model which deals with the electrophysiology, all the ion channels, which are pretty well understood in cardiac myocytes, deals with the ability to generate an action potential, the ability to handle proton bicarbonate exchange at the cellular level, the ability to handle the calcium transients in the cells and the control EC coupling that leads to calcium release and calcium binding to generate force. That's only just the beginning, really, in terms of useful physiological models, because all of those proteins are under the control of phosphorylation by beta-adrenergic signaling on a short time frame, but then lots of other signaling pathways that operate at different timescales to control the phosphorylation levels in those key functional units of coupled EC behaviour. So we've got to be able to deal with modules developed by different groups in order to be able to handle the physiology, the realistic physiology of these systems. So this is just to emphasize the fact that the three components really are the XML frameworks, the software that implements these and allows you to build models using these XML tools, but also allows access to the model repositories. So I think the whole concept of having a standalone software environment needs to be absolutely closely coupled to the model repositories. And we use all the standard Git type. So there's a version of Git encompassed in the software, so that as you develop your model, you can then deposit it back into the repository. And then another part of this, referred to here, is a framework called App Anatomy, being developed by Bernard de Bono. And this is where we want to be able to not only deal with the physics and have the frameworks for handling the mathematical physics aspects of the problems, but also access to all the anatomical terms when you're building, that you need when you're building an annotated model. So what Bernard has developed is this framework called App Anatomy that allows us to define all the physiology of the body. The epithelial tissues are shown on the outside, or invaginating here into the lungs. And then we can then dive into that. There's 50,000 terms embedded in that interface, so that you can then dive in and provide the appropriate linkages across different organ systems when you're dealing with diseases which inevitably involve multiple tissue types. So for example, if we're building particular models of transport, in this case of sodium, a very simplistic model, but you then have the ability to pull out the relevant cell-ML models dealing with transport processes in the context of knowing the anatomy and all the details of the anatomy that you want to embed those models into to build a more complex model. So here's another one for the kidney, dealing with transport from the renal tubule through to the capillaries. So where we're heading with this is we're setting up a journal that's going to be called the Physiome Journal, and it will be under the auspices of the International Union of Physiological Sciences, and it will be a secondary journal, not a primary journal. So when you send your paper to the normal peer-reviewed publishing journals, whether it's open access or not, we're working with those journals now to allow people who've developed models to be able, for the models to come to us so that we can curate and annotate them and make them available at the time of publication of the primary paper. And the idea, so we're working particularly with the American Physiological Society so that all their journals can make use of the same common framework, and we hope a lot of other journals. But the key to this is that the authors will get both a citation credit for their primary article, but will also get a citation credit for putting the effort into properly helping with curating, annotating and documenting the model. So they're getting a publication that will have an impact factor for that effort, as well as the effort that went into the primary publication. So we hope that that will provide the stimulus needed for more people to put the effort into making their models reproducible through these frameworks, but we'll see. So part of the effort is also now saying, if we have this large database of models that are properly curated, annotated, modular, can we pull them into a framework where we can begin to build physiological function on an algorithmic basis from these component parts? So we have lots of models of processes at this level, and then including 3D structure when we've got to the physiological level, and then assembling these into models of particular organ systems that then can be part of a growing database that deals with the whole human, so that we can study disease processes where those where the disease is going to have an impact on multiple types of tissue. And then I think the other important part of this is to link to the patient databases. So using the open electronic health record environments to be able to bring some of these physiological models, particularly with model reduction algorithms that allow them to be used in a clinical context, but to be able to bring them into the patient and clinical environment and coupled to patient records, so that we provide a more scientific way of recording the underlying data for a particular individual that can then help with the diagnostics and therapy for that individual. So there's quite a big effort now to build these frameworks and link them into open electronic health records. So I'm going to stop there. We've lots and lots of people involved in this work over a long period, but the particular work I've talked about for the cardiac work, particularly Martin Nash and colleagues, and on the experimental side, Bruce Smale, who I've worked with for many, many years, and colleagues on developing both the experiments and the instrumentation, and then the people involved in helping develop the Physiome project infrastructure. And then I talked a little bit about Merrin and Tor's work on the lungs and musculoskeletal modelling. So thank you, and just a reminder that New Zealand is very similar to Norway in many respects. I mean, we have a lot of snow-covered mountains, beautiful forests, and a long coastline like you do. We've got about the same size population. We're both long, thin countries next to much bigger neighbours. So there's actually a lot of synergy, I think, between New Zealand and Norway. So thanks very much.
Thank you very much. I think that we have time for questions. There's one down there.
Thanks, Peter. It's a great talk. Yeah, I think the CellML project in particular, at least within the cardiac field, for those people who aren't in it, has really changed the way that models are developed and the standardization of them, and it has made it a lot easier working in that field. One of the things that seems like it needs to be added is to link the models to function more clearly, not necessarily by a field ML or similar sort of application, but by annotating the models for what data they've been used and built from. And it's always been a bit of a mystery to me why that isn't a feature of most standardization programs.
Yeah. I totally agree, and one of the efforts that's going on at the moment in Auckland is we've been going through the CellML database and looking at where particular models depend on what data sets. So trying to annotate the provenance of parameters, because it's basically, when you look into any model, you'll often find that parameters are coming from multiple species and many, many different labs. So we've got to get a lot better at creating databases that properly recognize how parameters have been arrived at from particular experimental work and particular species and the conditions of those experiments. So I think the journal will be focusing on handling data repository as well as models, but only data that's associated with a model. And we will begin to try and implement more stringent requirements around how you annotate the data in relation to the model in order to address those issues. But it's absolutely vital, I agree.
We have time for an additional question. Yeah.
Thank you for the talk. I was wondering, for the secondary journal, how are you going to maintain? Is it by the fees that will be paid by the authors?
Yes, we're looking at an APC, an author processing charge of about $300. We thought, so we've got philanthropic funding behind the effort, but what's important, I think, is, I mean, the problem with open access is that the normal $2,000 fee, no one's going to want to pay that for a second article. So, but we thought that somewhere in the vicinity of $300 is going to be enough to sustain, given the other sources of funding we have for the infrastructure, sustain the journal, pay for the costs of helping a lot with the curation in particular. And maybe we can reduce that in the future as people get better at doing the curation themselves. But to start with, We thought, you can tell me if you think it's acceptable level, but I think an APC of that order is probably, given that you're getting another paper, a citation, is probably an acceptable level for the secondary paper.
Yes, thank you. Thank you very much. And here is a small token from Digital Life. And thank you very much. The next speaker is Dr. Jon Olav Vik, and he's at the Norwegian University of Life Sciences, or NMBU, and he is heading the project called Towards the Digital Salmon from a Reactive to a Preemptive Research Strategy in Aquaculture, DigiSal in other words. The title of your talk today is A Purposeful Simplification of Reality. Please, Olav.
Thank you. Yep. So, thank you so much for being allowed to speak, and thank you so much to Peter Hunter, who has laid the groundwork for a lot of what we aim to be doing in this project. But today I will talk a little bit about modeling more in general. So biological data is now being produced in such amounts that it can only be fathomed through the use of mathematical models. For a long time, biologists could complete their careers without mathematics and with very limited statistical training, relying instead purely on mental models of how biological systems work. On the other hand, theoretical biologists were usually so limited by available data that they could live their lives relatively sheltered from confrontation with empirical fact. And this is now changing under the omics revolution, and neither experimental nor theoretical biologists can ignore the opportunities for large-scale confrontation of biological theory with empirical data through mathematical models. Missed a beat there, but this illustrates lots of data. So let me wax a bit philosophical and ask, what is a model and why is it useful? Here we have something that looks to me a little bit like a turtle, or maybe a toddler baby crawling. But if you raise the head a bit and you stretch the neck a little, you have something that looks a bit like a llama. And if you stretch it further, you can approximate a giraffe. And what you can see from this very simple exercise is that making a model of a class of systems highlights both what they have in common, the overall form of the equations or the components of the figure, and also how they differ through the various parameter values. So model parameters are a good way to compare systems within a certain class of things. The project I lead in Digital Life is called the Digital Salmon. It seeks to understand the living fish body as a set of components that both affect each other and depend on each other. And this is what we mean by systems biology. And through this approach, we can draw more powerful deductions from experimental data than if we simply viewed the input-output relationship as a black box. So we look, for instance, at the mechanisms underlying the feed input to fillet output relationship. And the way to account for this is through mathematical models. The digital salmon will basically be imitating the virtual physiological human, which is Peter Hunter's life's work. So we want to make a knowledge base for Norway's most important biological resource, namely salmon, where we describe the physiological processes in the salmon body in mathematical form, and we link it to omics data of every relevant kind available. And this will be of help to the salmon farming industry in dealing with challenges of shifting feed prices, sustainability issues, disease challenges, and product quality. But then we had to decide where to start among all these interesting and important questions. And having thought about it long and hard, we chose to start where we can both manipulate, measure, and model. Because if any one of these is missing, it's going to be really hard to make progress. Experimentation is crucial in going beyond correlation to establishing causality. But to understand mechanism, we must be able to measure relevant aspects of the underlying processes. And without the mathematical framework to structure this in, we will be incapable of dealing with a sheer amount of information. So we chose to start with the challenges of novel feedstuffs in relation to the metabolism of salmon, where we can vary feed input, either in live experiments or in lab culture of tissue slices. We can measure thousands of directly relevant variables for the metabolic network through gene expression and metabolomics. And there is a very well-developed mathematical framework for describing and analyzing this through flux balance analysis and related approaches. So to recapitulate, the current challenge in feeding salmon has to do with the fact that it's not eating the same in captivity as it eats in the wild. In the early days, they were fed in captivity a diet based on fish meal and fish oil, but nowadays they eat 3/4 plants, both in terms of protein and in terms of fats. And it is an ongoing challenge to compose recipes for the salmon feed that are both cost-effective and cover the physiological needs of the salmon. It is also not very much more sustainable to feed salmon on plants, because those plants could have been eaten by people. So people are trialing new feedstuffs based on things such as yeast and microalgae and various other stuff I can't recall at the moment. So we collaborate with the Center for Research-Based Innovation called Foods of Norway, which is trialing. There they are, yeast, bacterial meal, microalgae, and doing this in salmon and also in other domestic species. So this is a hard optimization problem, and you don't have the time and you don't have the money to try all of this out in live experiments. So we need an in silico way to screen lots and lots of alternatives to keep or to filter out the plausible alternatives and to be able to rule out very quickly many things which theory shows us cannot work. So to look a bit in more detail at the metabolic network, this is a skeletal structural formula of the kind you may have seen in school, and it gets transformed through reactions that are catalyzed by enzymes that are coded for by the genes in the body. So basically everything you are is made from stuff you ate. And this building up and breaking down according to the body's needs is continuously ongoing. So if we have one of these reactions here from the breakdown of sugar, we see it's only one of several in the pathway for breaking down sugar. You can recognize the G3P and the NADA and the PI. And here you have a different representation of the same reaction in this graph. Molecules go in, molecules come out with the help of an enzyme. And yet another representation of the same piece of biological knowledge is in this matrix, where there is one column for this reaction, and it tells us that one molecule of H+ gets produced, one molecule of 1,3-DPG, one molecule of NADH. And this gets consumed. And you can do a lot of powerful computation on this, because it's just an account of how many molecules go in and out, and these are exact numbers. So if you look, for instance, at the rate of change for ATP over time, it is simply the sum of the rates of the different reactions that consume or produce it. And taking this stoichiometric matrix, multiplying it by the vector of fluxes, and saying that we would like to look at the case where this is zero, where you have flux balance, which is roughly true much of the time. And then you are left with the most studied equation in linear algebra. So you can automatically compute all the possible flux patterns that are consistent with equilibrium, and then you can begin to add more data to it. You can take gene expression data, where you see that not all the genes are expressed in all tissues or under all conditions. And you can then recompute what are now the possible flux patterns. You can take metabolomics measurements of how much you have of each molecule and further constrain the set of possible realities inside this system that is consistent with observation. So it's a very powerful home for a lot of data that we can now obtain pretty efficiently. So to summarize and generalize a bit, a model is a purposeful simplification of reality. It is designed to imitate some phenomena or behaviors or characteristics of a system, while downplaying the non-essential aspects. And this is often unappreciated by people who haven't tried to use mathematical models. You think, oh, I'm going to model salmon, or I'm going to model humans, but you're not really modeling the system, you're modeling what it does, how it responds to changing conditions or changing inputs. And the value of this lies in the ability to generalize from the model to a whole class of systems. So in this way, a mouse can be a model of mammals in general. An isolated heart cell in a petri dish can hopefully give generalizable insights about what goes on in the intact beating heart. And a set of differential equations describing how transmembrane voltage changes over time as a result of ion channels and the currents through them can give insight about the dynamics of the heartbeat. So as I mentioned in the example with the turtle and the giraffe, parameters are very concise and relevant phenotypes. So you can take thousands and thousands of variables, estimate fit mathematical models, and then take the parameter estimates as new phenotypes that give a comparison between systems from the viewpoint of the purpose of the model. This is also a very good way to combine knowledge from many disciplines and many experts into a functional whole, which is predictive and which facilitates the communication between us. Peter mentioned the importance of using common standards, so it is possible to plug together pieces of biological knowledge. And I very much sympathize with that, and I'm grateful that we have become a partner in the so-called FAIRDom initiative. So this used to be called semantic interoperability through ontological annotation, but nowadays they call it findable, accessible, interoperable, reusable. It's only fair. So it is about the same kind of thing, using the same vocabularies to annotate our data sets, using the very same vocabularies to label the variables in our models, and then you can let knowledge connect in an automated fashion. And of relevance to the brain modeling and nerve modeling people in the audience, your systems are mostly interesting for how they respond to stimuli. So you study them experimentally, you do things to them and you measure how they respond. And then you would want to have experimental protocols described in a reusable manner where you can apply the same protocol to a whole suite of candidate models for the same phenomenon. And designing standards that facilitates this is going to be very important for the large-scale medical use and food production use of these models. We need it for quality assurance, for documenting that the models do what we promise they do, and for checking that we're not breaking anything when we think we're developing our theories further. So, to reiterate, the digital salmon is meant to become an inclusive knowledge base. We want to give credit where credit is due and invite other people's work into this, using common standards so it becomes easier to build upon and to synthesize the knowledge that we acquire. This is a project with many partners. I would especially like to mention Stig Omholt, who is sitting there, and Arne Juvsland. Are you still here? Yep, there he is, who were also among the chief architects behind the vision of the digital salmon. So we will be revisiting Stig when this starts to gain traction and we're going to do serious business. Thank you.
Thank you, Jon Olav. We have time for one question. Back.
Thank you for the presentation. Concerning the FPA, is that based on a salmon genome?
Yeah.
I'm wondering, in humans, for example, we can't degrade, I mean, we do degrade complex carbohydrates, but the genes for the degradation is in the bacteria that is in our gut. So looking at back, you're going to use the FPA to consider which food or feed to use. Would it be an idea to include not just the salmon genome, but whatever it lives within salmon?
Yes. So I am aware that there has been done work in humans looking at how the gut microbiome is linked to the metabolism of humans, and they focus then especially on various kinds of vitamins B. So we are aware of the issue, and we have a roadmap of processes that we want to incorporate into this. We just chose to start where it is pretty certain we will get quick payoffs and where the road has been paved for us. But we definitely want to incorporate what you said.
Okay. Thank you very much, Jon Olav. Here is a small token from the DigitalLife. Thank you. Okay. Last speaker today, Professor Anders Goksøyr from University of Bergen. Anders is heading the project called Decod 1.0, Decoding Systems Toxicology of Cod. And that's your title today as well. Yes. Please. Yeah.
Thank you very much. And thanks for giving me the opportunity to talk here about this project. So we are staying in the sea. We are staying with fish. I will talk a bit about why we are doing this project, the approaches that we use, so how we are doing it, and also a bit on who we are. Results, we are still early in the project, so don't expect too many results. But as a former project, we are in the sea, different from salmon. Cod and other marine organisms don't live protected in a pen. They are exposed to the big ocean, the marine environment, and we know that the marine environment is not what we thought it was. It's not a pristine environment. Sometimes whales end up stranded along the coastline, and when we open their stomach, we see what's inside. Actually, this goosebeak whale that stranded outside Bergen in January had 30 square meters of plastics inside, and apparently this was why it was disoriented and stranded on the beach. So there is a lot ending up in the ocean, and what we want to do in environmental toxicology is to understand what happens, not with big plastics though, but with smaller molecules, when they enter the organism, so to try to reach a mechanistic understanding of what happens at the early stage, what this initial reaction leads to in terms of networks and chains of reactions inside the cells and inside the organs, and also how we can extrapolate this into understand what happens at the ecosystem level. We also have an interest in evolutionary and comparative aspects of these of the systems that are responding to pollution and environmental stress, and we also think we can use some of this knowledge to develop tools for environmental monitoring and human health understanding. So we are also talking of different levels of complexity, different levels of mechanistic understanding, and this is where we need new tools to develop our understanding. This is where systems biology, or in our case, systems biology comes in. And the approaches that we use is to work together with different fields of science, so transdisciplinary approach. We have a large group of people. We gathered almost 25 people at our workshop at Voss just before Christmas. People from experimental side and from the computational side, from the mathematical side. Our project has 9 work packages, so we try to develop or produce data in the initial levels, produce samples by experimental studies, analyze these samples through chemical, genomic, and physiological analysis, and then to integrate the bioinformatics and the mathematical people into these data. We're also looking at biomarker technology development and, of course, the management and ROI issues. And I will return to some of this in this talk. So the cod, why are we looking at the cod? It's a widely distributed species in the Atlantic Ocean. It is an important fishery species. It has become more and more used in environmental monitoring as an indicator species. And we are building a basis for using it as a model in toxicological studies. This is, of course, very much based on the sequenced genome that was first published in Nature 2011. Kjetil and other people involved in that. And there is a new assembly of a deeper sequencing of the cod genome that is now available, at least to the collaborators in the project. So the experimental approaches that we use is to expose fish in tanks, is to work with liver slice cultures, and it's to work with isolated proteins, receptors that we recombinantly express and produce reporter assays where we can study their responses to specific contaminants. So from these types of studies, we are developing a basis of data on through genome mining, transcriptomics, oops, that was the wrong one. Through genome mining of the cod genome, looking at what are the complementary genomes that are present in genes that are present in the genome, the transcriptome produced through different exposures, the proteome and the metabolome, where we initially at least will want to focus on the lipidomics part of the metabolome. So an example from these precision cut liver slices that we also use, similar to Jon Olav's talk on the salmon. With the cod, the problem is that the cod liver is such a fatty organ. It's at least 50% fat. So isolating primary hepatocytes has proven to be very, very difficult. They tend to destroy through the process, and we lose almost all of the cells. And so as an alternative, we have developed this precision-cut liver slice system. It's a bit different from the salmon system because of this fatty tissue, but isolating cores, molding them with agarose and slicing them with this vibratome produced then tissue slices that we can expose and use as our experimental systems. And so it also saves a lot of fish. We are reducing the number of live fish used in experiments. And from exposing liver slices, we can produce data from individual compounds where we produce dose response curves through compounds like benzopyrene, PCB. And we know that we have a target receptor for these compounds in the dioxin receptor or AH receptor. And producing a luciferase-based reporter assay, we can compare the data we get in the liver slices to the individual receptor systems. And we can do this for many different compounds, for different receptors, and produce data sets of responses to individual compounds and to mixtures. And we then move to isolating these receptors that we know respond to specific sets of contaminants. And we want to use those as biosensors for detecting these contaminants in the environment, in environmental samples. And so from using them in a luciferase reporter assays, we can clone and make recombinant proteins that are used in a cell-free system, in the alpha lysa system, and hopefully we can move into a lab-on-a-chip system, which is one of the other projects in DigitalLife is focusing on, where the receptor is the functional molecule detecting compounds, contaminants in the environment. And we have posters on this and the other parts of the project afterwards, so hopefully you will have a look at those. Some of the data we have been able to generate is based on previous experiments we have done with exposing fish to single compounds. And from these we have generated pathways that we know or have seen responding with different genes and proteins in the pathway. This is the fatty acid biosynthesis pathway. And so we know that our target genes that we want to look further into, these are the differentially regulated genes or proteins. But we're also looking into using the cod as a canary in cage in the environment. And this is one example, a video I would like to show to you.
For over 40 years, all waste from Bergen was dumped in Kollevågen, off the coast of the island of Goksøyr. In 1983, the area was reopened as a popular beach and recreational site. However, in the last few years, old sins are reappearing. We want to find out how this situation is affecting the environment. We have kept cod in cages in Kollevågen for 6 weeks. Using an ROV to transfer live video to a laptop, we've monitored the well-being of the fish during the whole experiment.
So we had 2 days of sampling where we sampled almost 90 fish from 4 different stations. About, I think it was 13 tissues from each fish. So more than 1,000 samples were collected during these 2 days. And some of them were processed directly on board the ship.
Samples are now safely stored and awaiting further analysis.
And some analysis is now actually being processed through qPCR and waiting for RNA sequencing at the sequencing laboratory. And so from these types of experiments, we, of course, generate enormous data amounts. Our computational biologists, mathematicians, and bioinformaticians are very impatiently waiting for these data. They feel that we don't have enough data at the initial part of the project, but we hope to be able to make them happy soon. And so this is where the next work package is, where we integrate bioinformatics and mathematics into the project. And the teams are working in the group. We have good cross-disciplinarity, both in the mathematical sense, so different groups at the mathematical department are working together. We try to develop a multidisciplinary language. In this case, we used our workshop to have the other discipline trying to explain what they didn't know to the rest. So kind of a biology for dummies by dummies. And it seemed to work. And of course, within the mathematical group, we are working on dynamical modeling, building models that hopefully also can both explain responses, but also assist in experimental design. And in topological data analysis, we try to use the technology to explain and decipher patterns in the big data masses. And actually, the mathematicians here have already developed a new method, new softwares that can be used to improve the efficiency of TDA. So there is also innovation going on in this field. The last work package is, of course, the management and RRI. We have a coordinator in place, Marta Eide. She's now on maternity leave. We have a steering group. We have a website. And we have RRI activities coming on. Dorothy Dankel has been hired into this project to work coordinating RRI activities. And one of the maybe most, at least forthcoming activities is the Fish Hackathon that will take place in end of April in Bergen, where modelers and app developers, designers, students will meet to try to develop apps for sustainable fisheries, for contaminant monitoring, for seafood safety, etc. And so this is also a big project with many partners. These are a list of the collaborators, the different institutions and names. And we have, as I mentioned, the website where you can check for further information and activities. Thank you.
Thank you very much, Anders. We have time for a question, and I saw an arm here.
So I have thank you for the talks. I have a question about that is organizational. So you say the bioinformaticians are eagerly waiting for data, and I know this feeling, and in a collaborative project, you know, this is always a bit of a problem. So how do you see this in projects where we have, you know, we heard it, we are limited by time? How do you see this? You know, you want to gather data, and then, you know, somewhere the bioinformaticians have to start analysis, and how do you solve this?
How we handle it? Well, I think we have handled it with two approaches. One is we've had some previous projects which have maybe not as extensive in scope, but there are some data sets that are available to use. We also know that there are databases with experiments not from cod but from other fish, from mammals, that has not been exploited properly, so there is plenty to do on available data sets within toxicology. But we also know that data sets from the medical field, from other fields of biology, could be training sets also for the approaches that our mathematicians want to use. So we don't want to limit them to sit and wait, but also encourage them to look elsewhere for data.
Okay. Thank you, and thank you very much, Anders. Here is a small token from DigitalLife. This was the last talk here today, oral talk. Next on the program, besides the coffee break, is of course the poster session. And we're a little bit, let's say, later than scheduled. So I think that the poster presenters now should try to put up their posters. The poster session will be where we had the lunch today, I have been told. That's correct. So I think that the poster presenters' task now is to try to put up their posters. We'll see how long that takes, and I think that the easiest would be that we give a sign when the poster session starts. And we will also arrange it in a way that each of you will get a chance to present very briefly your poster. Okay, and thank you, all of you, again.